Hey, so today's amino acid is serine, right? So SCR is the three-letter code, and S is the one-letter code. And so let's look at that. Here's our alpha carbon with the amine and the carboxylic acid on each side. Here's the R group. Now the R group of serine has a beta carbon and a hydroxyl. Right? Now this hydroxyl is pretty reactive, right? It's a precursor for a lot of essential biomolecules. This includes the sphingolipids, phosphatidylserine, even glycine, as well as cysteine. Cysteine actually is very similar to serine, but it replaces the oxygen with a sulfur atom, right? Now the hydroxyl group makes it very reactive, very highly functional. This can act as a nucleophile. It can form hydrogen bonds, so it's a polar functional group. It accepts phosphate groups, and it's very important for enzyme catalysis. For example, in the serine proteases, this is the first enzyme in the catalytic triad, right? And this is what actually has the catalytic activity of serine proteases, such as trypsin, chymotrypsin, and elastase. Trypsin and chymotrypsin, of course, very involved and very useful in digestion. So that's very important there. Now, it's also important in cell signaling because this hydroxyl means it can accept and react with a phosphate, inorganic phosphate, and that can serve as kind of a tag or an on-off switch, or maybe even a dimmer switch, if you will. That's very useful in signal transduction, enzyme activation, inactivation, regulation, even in cell cycle control. So serine is a very important molecule for hormones, for nutrients, and also involved in the stress relief pathway. So there is serine.